Okay guys, so here we are. We're going to run through the steps of muscle contraction again, kind of make sure that you're clear on all the pieces, okay? So starting off the bat, remember that this whole picture is a muscle fiber, skeletal muscle fiber. And remember that that word means that this is a skeletal muscle cell, okay? Within there, we have some extra vocab, like the sarcolemma is the cell membrane, the sarcoplasm is the cytoplasm. But the key parts end up down in here with the myofibrils. So remember, the myofibrils are each one of these tubes. So here's one in the middle, and there's another myofibril, another one. And those myofibrils have within them the actin and myosin. That's what we're really going to be focusing on. Okay. In terms of the big picture, remember that wrapped around each myofibril, you also have this sarcoplasmic reticulum, everything marked in blue here. Okay. And the sarcoplasmic reticulum is filled with calcium. Okay. And that calcium is going to be really important in the muscle contraction itself. And then the last big piece we have here are the T-tubes, which allow the impulse to get to the inside. So when we look in, if we zoom in, remember that these blue stripes inside here are myosin. Okay? And that's what we're actually looking at here. So each of these blue stripes is myosin. Each one's 296 of these little small proteins, okay, and the key portion of this protein is this head, which is able to bend. So this is it'll bind, hold, and bend, and that bending actually creates the force for our muscle contraction. So hundreds of thousands and millions of these little heads bending is what generates the force when a muscle actually contracts. Right? And what is it generating the force on? Well, these heads have to bind onto the red portions in there, which are shown here in this next line. Okay, so these red portions, if we zoom in on them, most of the red portion is actin. That's what all this orange is in here. It's actin. Okay, and in fact, a lot of your classes, people will just talk about myosin and actin. They won't talk about the other parts, but these other parts are really important for us. So remember that on the actin, you have those gold binding sites or active sites. I often call them binding sites. And that's what those myosin heads are trying to grab hold of, and they're blocked off so they can't really do their job. Right? So that's the function of the tropomyosin and the troponin. Okay, and these two parts control whether a muscle is actually going to contract or not. If the tropomyosin is in this state, covering up the active sites, the myosin can't grab hold of the actin, so nothing's going to happen. But remember that the general way it works is calcium is going to come in here, bind to that troponin, making this all change shape and move out of the way. So therefore, we'll actually get the contraction. Okay. So this altogether is what is often called the thin strand, because looking at this picture up here, you can see that the red is thinner. Okay, and the purple is often referred to as the thick filament because it is much thicker. The protein itself is much thicker. Alright, so when we decide to move, remember these are skeletal muscles, so they're all voluntary muscles. So the signal is going to come down from our brain, goes down, goes out a motor neuron, and we're going to get impulses here. Because we have myelin on here, this is going to be saltatory conduction going down. We get action potentials in each of those nodes. But here comes the signal coming down, and when we get to here, this is a synapse. The neuromuscular junction is a synapse. So there's a small gap, and you can kind of see it depicted pretty well down in this little picture here. Each of the red dots would be neurotransmitters. We only have to worry about one neurotransmitter, ACH. It's the only neurotransmitter with skeletal muscles we worry about. When that gets released, it's going to go across and bind, and when it binds, we're going to get an impulse. So imagine over the surface, there's a whole big electrical wave going over the surface of this muscle fiber. Okay. Now, over the surface, it would only stimulate these myofibrils on the very outside. Okay, maybe these guys too over here. But remember, it also can dive in through these T-tubes. Here's one of the openings of the T-tubes. Here's another one on the side here. So the electrical signal goes in here all the way through this like fine network of gold channels in here, and that means that the electrical wave goes through the whole thing. Okay, and remember the key to the impulse. The impulse is going to let in a tiny bit of calcium. Okay, so a little bit of calcium from outside the cell is going to come rushing in. Remember, most of the calcium is outside of cells in the body. All right, and that little bit of calcium coming in is going to cause these sarcoplasmic reticulums. Remember these guys here. Okay, or the sarcoplasmic reticulums, they're filled with calcium, and that's what floods this entire thing. So what essentially happens is you get the action comes down, you get the synaptic transmission where we release the acetylcholine, that binds, causes this impulse over the surface, the impulse goes in through the T-tubes, that lets in a little bit of calcium, which causes all the sarcoplasmic to release calcium, and the whole thing gets flooded. This whole area is now flooded with calcium everywhere, okay? Alright, so all that calcium gets flooded in there, alright? The end result is kind of looking at the edge. So let me go back for 1 second. If you turn this around, this little piece right here is what we're going to look at. So if I flip that around and turn it on its side, that's what you're seeing here. One myofibril with all the actin and myosin inside there, okay, with wrapped around it the sarcoplasmic reticulum. So if you zoom in on the sarcoplasmic reticulum, which is what we're doing here, you see the calcium being released. Okay? So at rest, take a look at the picture, at rest, this cross bridge, this arm, can't reach the binding site because the troponin and the tropomyosin are in the way. Right? But as soon as the calcium comes along, that's going to bind to this troponin and the tropomyosin. And remember, it's a protein, so it changes shape when you get a positive charge binding to it. Right? So it moves out of the way, and what do you know? Open, wide open here. The myosin cross bridge is now has open access to the binding sites, and it's going to grab hold of it right there. Okay, so the calcium coming from the sarcoplasmic reticulum changes the structure of the troponin and the tropomyosin, moves them out of the way, so the myosin can now grab the edge of it. Right, and then from the side, looking at the exact same thing from the side. Okay, covered up because we're at rest. Okay, so it's covered up, so there's no room for binding, but here comes some calcium. They change shape, and look, they move. So now the binding sites are open, and here comes the cross bridges. They can now grab hold, and that's what we see in this picture. Binding. The sites are exposed, so they grabbed hold. Those two spots have grabbed hold. Okay. You'll notice on top of them ADP and P. That used to be ATP. That was broken apart. So when that ATP is broken apart, that gives us the energy to bend the heads and move the heads. So now we actually have a muscle contraction taking place. These heads are actually bending and moving the actin inward, pulling the actin inward. Right? And in the end, we also need ATP to let go. So we need a new ATP to let go, and then we break it apart to reset the head. Okay, so now it can do it again. And if we're contracting, if we're moving, a glass to our mouth like we've been using in class, or we're picking something up to drink, you're going to do this over and over. It's going to bend, new ATP, let go, reset, bend again, and you'll get this kind of whole cycle repeated over and over and over in the course of a contraction. Okay, so this is now reset, and the very next thing that would happen was, if there's still calcium, grab hold again, and a new bending, moving the actin even further in, and same thing would happen here. This would bind and then bend that way again. All right? So the idea is, when we're all said and done, at rest, notice this big gap in here in the middle. Okay, you got this big gap where there is no actin in the middle, but in this one we've pulled inward. Okay, and remember that there's another chunk right here. This whole section is called the sarcomere. There's another chunk over here doing the exact same thing. This part just got shorter, and this part just got shorter, and all the way down the line of the muscle, so the whole muscle gets shorter as it moves. Alright, so these are the questions I want you guys to do tonight. There's just 5 of them. Just write them on a separate piece of paper. First one's true or false, get you off to a nice start. But really think on them, really try and put them in complete sentences, guys, and this will give us a good indication on whether you really understand what's going on in muscle contraction. Alright, so good luck, and see me tomorrow if there's any questions on this, guys, because this is where the rest of our unit comes off of these ideas. Alright, good luck.